Coffee kickstarts our day and powers us through the night. Globally, coffee is consumed in vast amounts with the 2.5 billion cups enjoyed daily.
We at MIT-MAHE identified that one of the world's favorite flavors is under threat.
Coffee leaf rust, or CLR, is a major fungal disease caused by Hemileia vastatrix that severely threatens coffee production, especially in the Indian state of Karnataka, where coffee plays a crucial role in both the economy and heritage. The impact of coffee leaf rust on crop production can be severe, often reducing yields by 30 to 80% depending on the severity of the infection and the effectiveness of management practices. This devastates coffee production and impacts millions of farmers worldwide. In regions like Karnataka, where coffee is a primary export, these losses significantly affect both the local economy and global supply chains. We got the opportunity to talk with Dr. Athena Couturius from the University of Copenhagen, who helped us understand the fungi's modus operandi. The fungus begins by adhering its uridinospores to the underside of coffee leaves, where they germinate and form aprosoria, specialized infection structures that enable the fungus to penetrate the stomata. Once inside, the fungus produces haustoria, which are responsible for extracting nutrients from the plant's cells. As the infection progresses, the fungus expands through the plant's tissue via intercellular hyphae and haustorial mother cells, weakening the plant. The fungus secretes effector proteins that suppress the plant's immune response, allowing the disease to spread further. Eventually, the fungus produces more spores through sporulation, perpetuating its destructive cycle. We identified key antifungal compounds having antagonistic effects against coffee leaf rust, specifically those that disrupt its life cycle, and thus solidified our project's plans, presenting CAAPI, Coffea arabica protected by Iturin.
Iturin A is a broad-spectrum antifungal lipopeptide produced naturally by the Bacillus species present in the coffee plant microbiota. During our search for Iturin A producing strains, we developed an Airtable, a comprehensive repository of Bacillus strains. With over 100 strains catalogued, it includes detailed information like each strain's ability to produce 3 important lipopeptides: iturin, fengisin, and surfactant. We've also included supporting literature and strain accession numbers for easy lookup. Among all lipopeptides, Iturin A has the strongest antifungal capabilities. It induces systemic resistance, which has proven to be effective against Hemileia vastatrix. It kills the fungus by disrupting the integrity of its cell membrane, leading to the leakage of cellular contents and eventual cell death. It also induces oxidative stress within fungal cells, causing further damage to cellular structures. This lipopeptide is synthesized through non-ribosomal peptide synthesis modules formed by the enzymes translated by the Ituoperon. This operon consists of Itu-D, Itu-A, Itu-B, and Itu-C. The gene Itu-D encodes malonyl-CoA transacylase, which plays a critical role in fatty acid synthesis. The genes Itu-A, B, and C encode for enzymes responsible for initiation, elongation, and cyclization of this heptapeptide.
The Iturin operon's native promoter P-Itu does not produce enough Iturin A to kill Hemileia vastatrix. The Iturin produced by our Bacillus strain ATCC13952 was quantified through HPLC. We conducted 12 runs with various concentrations of the recommended solvents, namely acetonitrile, 0.1% TFA in water, and methanol to build the ideal HPLC method file. With the help of Nayana Subhash, a research scholar at MIT Manipal, we developed the final HPLC method file for our Iturin A, which is as follows. The graphs obtained as a result depict the Iturin concentration produced by cells grown in LB and PDB. As we can see, the Iturin produced is low, confirming that P-Itu is a weak promoter, prompting us to explore strategies to enhance Iturin A production. We decided to opt for swapping out P-Itu with a stronger promoter using homologous recombination. Among all, we picked 2 strong promoters to test against each other, P-BAC-A and P43, both of which have proven to show enhanced Iturin A expression in Bacillus. P43 is expressed continuously, that is throughout the life of the bacteria, and shows high expression in the logarithmic phase. It is a sigma A dependent promoter. P-BAC-A expresses during the stationary phase when bound by the sigma factor B, which is a transcription factor produced in response to stress. This stress could be directly or indirectly induced by the fungus.
Why P-BAC-A and P43? Using a very strong promoter like CTUP will increase the metabolic load of the bacteria and may lead to mutations of the strain, compromising in Iturin A production. Hence, we chose a promoter that was strong enough to produce sufficient Iturin A to kill the fungus Hemileia vastatrix. MATLAB modeling was done to simulate constitutive and inducible gene expression of GFP with the promoters P43 and P-BAC-A using the data from literature. We were advised by Dr. Sabri to have about 500 to 1000 base pairs of homology on either sides of our desired insert. However, based on Bacillus subtilis, transformation with linear fragments suggested a minimum of 70 base pairs for plasmid integration with an ideal range of 100 to 500 base pairs for double crossover events. Given this information and to ensure our fragments are not too long so as to avoid self-complementarity, we designed the P-BAC-A fragment with 500 base pairs of homology and the P43 fragment with 350 base pairs of homology with GFP as a selectable marker to confirm integration.
We performed TCR to amplify both promoters for HR or homologous recombination. The amplification results showed P-BAC-A having strong amplification while P43 was weakly amplified, proven by the light bands. This can be attributed to suboptimal primer design, including issues like hairpin structures, primer dimers, and del-G values. We performed HR using the protocols provided to us by Dr. Sabri, which was optimized to suit our strain. The results were as follows. Green fluorescence was observed in the colonies that were transformed with P-BAC-A, indicating that the promoter was successfully integrated into the genome. This wasn't observed in the case of P43 due to weak amplification of the promoter as mentioned earlier. This was a key factor in proving our engineering success, helping us troubleshoot for further experimentation.
Along this journey, through a meeting with Dr. Aditya P. Sarnaik from Arizona State University, we were led to the idea that desirable features of the two promoters P-BAC-A and P43 could work synergistically when placed in tandem. This dual approach ensures optimized Iturin A production throughout all the phases of the bacteria's life cycle. Working around this idea, we received low synthesis scores for any fragment containing P-BAC-A from IDT. We tried to resolve this issue by deleting a set of repeats in the promoter's upstream region past -80 region. The -10 and the -35 regions of the core promoter where the sigma factors bind heavily influence transcription and any modification can better the process. These regions were substituted with the consensus sequence for sigma A and sigma B on P43 and P-BAC-A respectively. Meetings with Dr. Balaji from MIT-MAHE made us realize that the structure of the RNA polymerase influences its binding to the promoter region. He also suggested we take into the account the conformation of the sigma factors when it binds the DNA. We have realized that promoter engineering is a multi-component process. As a contribution, we have created the Promoters for Dummies handbook. It's a step-by-step guide for students and research on how to design prokaryotic promoters. We have broken down complex concepts like transcription, the different components of promoters and the essential steps in optimizing them for your experiment.
Over multiple meetings with Dr. Fayaz from Manipal Institute of Technology, we studied the binding interactions of our constructs through molecular docking and dynamic simulations. The software HDoc was used to dock and predict the binding energy between the promoters and an essential subunit of the RNA polymerase holoenzyme, the sigma factors, which are responsible for the recognition of the core promoter region. Docking helped us compare the optimized and unoptimized promoters. The results suggested that the unoptimized P43 had better binding affinity towards sigma A and P-BAC-A optimized had better binding affinity towards sigma B. To test the functioning of our dual promoters, we plan to compare it against the individual promoters P-BAC-A, P43 and PE2 in plasmids in both E. coli and Bacillus subtilis.
Our team prioritizes safety and security in all lab work, ensuring the well-being of members, environmental protection, and ethical practices. Working in a BSL-2 lab at the Department of Biotechnology, we adhere to strict biosafety guidelines. All members receive training in risk assessment, equipment use, and waste management. We follow strict safety protocols, including the use of personal protective equipment, regular sterilization, and proper waste disposal. Hazardous reagents are handled with care and potential risks from pathogens are mitigated. After engaging with multiple stakeholders who raised concerns about the release of GMOs into the environment, we developed a carefully designed kill switch as a safety mechanism. Our kill switch assures safety by replacing the natural promoter of the GAL-E gene with the QMate-inducible promoter in the Bacillus subtilis gene. This allows the expression of the enzyme that degrades galactose only in the presence of QMate.
Cumate is biodegradable and a non-toxic compound without which toxic galactose metabolites accumulate, eventually leading to the death of the bacteria.
Feedback from multiple stakeholders and industry professionals has been instrumental in shaping our approach to combat coffee leaf rust. Currently, 3 primary control measures are used: copper-based chemical fungicides, resistant cultivars, and agronomic practices like sanitation and shade regulation. After evaluating these methods, we developed an eco-friendly solution, a Bacillus subtilis wettable powder. This biocontrol agent offers high spore density, ensuring potent activity against Hemileia vastatrix. The wettable powder formulation allows easy integration into existing agricultural practices, providing both preventative and curative action against CLR. Key components include a genetically engineered Bacillus subtilis strain with a dual promoter system for Iturin A production, alongside carriers, dispersants, wetting agents, stabilizers, and UV protectants. To support the development of such formulations, our contribution, A Beginner's Guide to Biopesticide Formulations, offers a detailed overview of different formulation types. It highlights their strengths and limitations while providing practical insights for field applications. This guide empowers researchers and agricultural professionals to create sustainable, eco-friendly pest control solutions tailored to various agricultural needs.
The farmers and stakeholders we reached out to expressed their concerns about the coffee yield being erratic and difficult to predict. After a meeting with Maurice D. Moss, former iGEMer, we realized that farmers are at the heart of coffee agriculture. The software team wanted to contribute in ways other than just our Iturin spray. For this, we built a yield prediction model which we lovingly call BREW, Bean Research and Estimation Workflow. This model could help farmers and researchers alike to predict the yield during harvest beforehand, enabling them to make informed decisions. We achieved an R-square score of 0.986 while training and testing our model. Coming to the data set used, numerous meetings with Karnataka's Planters Association helped us validate the parameters that we used for the model. We have consolidated data for 15 parameters like bearing area, rainfall, and temperature district-wise from the year 2000 to 2023 from verified sources like Coffee Board of India, WRIS, NATMO, and NASA PowerTape. This dataset is also one of our contributions this year, as there is a scarcity of consolidated datasets with respect to coffee, which in some way is holding back the research scene in the coffee world.
The CAAPI project is grounded in human-centered design, a systematic approach that prioritizes stakeholder needs and feedback. This framework enabled us to identify, engage, and integrate the various perspectives of individuals and organizations critical to addressing coffee leaf rust effectively. We began by creating a stakeholder map which characterized our key stakeholders such as farmers, researchers, and regulatory bodies. This map helped us ensure that all relevant perspectives were considered throughout the project. Farmers in particular were identified as the primary stakeholders due to their direct experience combating coffee leaf rust with chemical fungicides. Engaging with them allowed us to understand the practical challenges that they face, such as dependency on chemical treatments and uncertainty in coffee yields. Discussions with the farmers highlighted 2 major issues: the limitation of chemical fungicides in managing coffee leaf rust and unpredictability of yield at the end of each harvest.
Upon interaction with our indirect stakeholders and consumers of coffee through our survey forms, we were able to gauge that it was our responsibility to educate the common man about not only coffee leaf rust, but synthetic biology itself. Our education efforts catered towards all age groups of students and working professionals. We used a variety of content including presentations, animations, and an audiobook to explain basic biology and synthetic biology principles. Our responsibility did not end with just education. It extended beyond just that. We hope that our efforts into alleviating coffee leaf rust inspire future teams to take up projects addressing the difficulties faced by farmers, especially for commercially important plants such as coffee.
Our journey with CAAPI is one in progress. We are presently working on quantifying Iturin A against Hemileia vastatrix and testing our dual promoter. Using this data, we are looking into commercializing a wettable powder formulation with the Bacillus subtilis strain present in the coffee plant microbiota. Our project was born out of our interactions with our primary stakeholder being the coffee farmers, and we want to give back to these who are involved in bringing a favorite drink to our table.
Last but not the least, we would like to thank all of our stakeholders, professors, and experts for guiding us through the entirety of our project and shaping it into what it is today, and for helping us building upon it even right now as we speak. And of course, thank you for watching.